My name is Kristen Twee. Oh, and I guess we're recording today, so sorry about that. My name is Kristen Tweel. I'm the Director of Sector Innovation at Genome Atlantic. I'll be co-hosting the session today with my colleague Janelle Scared of MICA and CNA. We're really thrilled to have everybody here. So we've got a fantastic lineup of speakers. Just one quick comment that unfortunately Dr. Sue Baldwin fell ill, so she thankfully is being replaced by Cassie Appelt from Kuhnke. So Cassie, thank you so much for joining us today. All three of our speakers, we're really thrilled to have them. There's a great mix of both industry and academic experience here. And before we kick off, we'd like to offer a land acknowledgement statement. We respectfully acknowledge the land on which we gather as the unceded ancestral homelands of all Indigenous peoples who were here before us, those who live with us now, and the 7 generations to come. As First Peoples have done since time immemorial, we strive to be responsible stewards of the land, to respect the cultures, ceremonies, and traditions of all who call it home. As we open our hearts and minds to the past, we commit ourselves to working in a spirit of truth and reconciliation to make a better future for all. Thanks. So as mentioned, my name is Kristen Tweel, and I work for Genome Atlantic. And if you're not familiar with Genome Atlantic, we are a not-for-profit organization that is part of a national network under Genome Canada. Our mandate is really to bring sort of the social and economic benefits of genomics, or as we like to call it, biotechnology approaches to different sectors that impact our region. So as we're an Atlantic organization, really our mandate is to serve all of Atlantic Canada, so Nova Scotia, New Brunswick, Newfoundland, and Labrador. Since our inception in 2000, we've enabled over $170 million in research. We do that in many ways, but one of the things that's really important to us and what we focus on is translational research. So we work on de-risking investment in private sector for research and development projects. Another one of the many facets of the services that we offer involves education. And one of these things you can consider like a webinar, where this is really meant to help educate the sector on the benefit of biotechnologies. More and more, we're seeing our funders, our government partners emphasize the importance of this mining sector to our region, looking to us for solutions for critical mineral extraction, remediation of legacy sites, and on and on. We believe that genomics is a tool that can really help provide more sustainable solutions to the current way of doing things. So hence the webinar. And I'd also like to highlight really quickly that as we're part of a national organization, the Genome Canada Enterprise, there are 5 other centers like us across the country. And so if you're in another region and you'd like to be connected to another genome center in your region, we'd be more than happy to do that. So I'm going to pass it over to Janelle, who will introduce herself and kick off with a little bit of information on the agenda.
Thanks, Kristen. Yeah, so my name is Janelle Scared, and I'm the Mining Network Development Officer for the Mining Innovation Commercialization Accelerator, aka the MICA Network. And I'm affiliated with College of the North Atlantic, who is the main regional partner for MICA in the Atlantic Canadian region. So in this role, I work to bring together mining companies and stakeholders with innovators and researchers from various other sectors. In a nutshell, we're constantly seeking ways to make the mining industry greener, lighter, more efficient, and more sustainable by integrating innovative solutions and technologies. And because I am affiliated with both CNA and MICA, I'm pleased to provide you with a brief overview of both of those entities. So the Mining Innovation Commercialization Accelerator Network is a $112 million pan-Canadian program that's led and managed by the Center for Excellence in Mining Innovation. MICA seeks to eliminate the gap between the conceptualization and the implementation of new technologies and solutions geared towards mining. And the College of the North Atlantic is NL's only public college, and it spans the entire province through 17 campuses across Newfoundland and Labrador. So as the MICA main regional partner for the Atlantic region, CNA brings to the network its top-tier mining research capacity, including faculty researchers, highly qualified personnel, hyperspectral imaging, and reality capture technologies, to just name a few. So I think we can move off to the agenda slide there, Kristen. Perfect. So while many of you here today might be familiar with some of the concepts that we're going to be discussing, the initial goal for this was to offer it as an introductory session to some of the ways that different biotechnology approaches could benefit the mining industry throughout the life cycle of a mine. And we really encourage following up. And as a mining company, if any of these approaches pique your interest and you would like to learn more, we certainly encourage you to follow up with us and the speakers. So, quick notes in terms of housekeeping. Today's session is really formulated as a series of short presentations followed by a panel discussion / Q&A session. So each speaker will have 10 minutes to provide an overview of their topic. And then at the end of those 3 presentations, we're going to open the floor to questions, which could either be directed towards one panelist or all of them. Or, maybe, you actually see a connection between 2 of them and you want to ask that. So we really envision this to be an engaging dialogue where we could leverage the expertise and the knowledge of those panelists to help attendees better understand how genomics could be applied to your operations. Questions can be submitted via the Q&A button. You should see that on the bottom of your screen. So when you submit a question, if you could please indicate who the question is for, myself and Kristen will be monitoring that function as questions are submitted, and we will then relay those questions to the appropriate panelists. So at this point, I'm going to turn it over to Kristen, who will introduce each of our speakers and their presentations.
Thanks. All right, here we go. So we are thrilled to introduce our first speaker, Dr. Mehrdad Hajibabe. He is an internationally recognized leader in the molecular biodiversity and genomics technology space, if you're not familiar with him already. He's an associate professor at the Center for Biodiversity Genomics and Department of Integrative Biology at Guelph. He's also the co-founder and CSO of a really cool company based in St. John's called eDNA Tech. I've had the pleasure of working with Mehrdad in the past and seeing him speak, so I look forward to seeing what you have to say today, Mehrdad. Thank you.
Thank you so much, Kirsten, and all the organizers. And when you say I have 10 minutes, I was just quickly removing the slides from my presentation. But hopefully I can convey the message about the work that we are doing. I'm gonna try to share my slides and we'll go from there. Just give me a nod there. So good. I'm gonna put it in the. All right, sounds good. So yeah, basically today I'm going to be talking about the use of genomics and namely environmental genomics approaches and environmental DNA to understand biodiversity in different settings and to use that information for environmental assessment. I'll get into some of the work that we have done that have relevance to the mining industry, and I have been engaged in sort of discussions with colleagues from mining for over a decade and participated in a number of conferences and sessions on the use of genomics in mining, and I'm glad to see sessions like this and some of the new approaches and updates that we have hopefully will make sense for the audience. So let me set the stage of by talking about biodiversity at large and the level of the organization. So this circular diagram is the tree of life, sort of the evolutionary tree of all the organisms. We don't have microbes sort of represented here well. They have obviously a vast diversity themselves, which mostly unknown. But we have animals, fungi, plants, and protists. Now, we have been organizing these into various functional sort of groups in terms of studying biodiversity and making use of that information. I'll try to sort of organize it here. For example, like we have endangered species, species that the populations are under threat for various reasons and impact of various types of activities, like examples like marine mammals, some of the amphibian species and so on, and then we have on the other side sort of invasive species that are sort of harmful to the ecosystems, and usually they're transferred through human activities from one ecosystem to the other and can invade and they damage the ecosystem there. And then we do have organisms that we use to, we call them bioindicators. We use them for identifying and understanding ecological health and changes. And then pests, pathogens, and food and natural health products, commercial species, and so on. So all of these need to be tracked. And interestingly, and or maybe the big challenge there is how to track them across time and space, because it's one thing to discover organisms and understand their diversity and sort of classify them. Is another thing to be able to track them and monitor them. So and then we do need technologies and tools that are robust enough and feasible to be able to help us monitor all of these different groups of organisms. And then there is, like, in the past 2 years, especially, there has been a lot of demand for biodiversity data, and that, unfortunately, that's probably because we are dealing with a crisis in terms of species decline and extinction. And like with we see a lot of publicity around this from various organizations. We had the global biodiversity community sort of are now sort of moving towards incorporating various types of regulations in terms of understanding the impact of human activities, and on biodiversity, and using measures or developing measures and tools that can, help companies and organizations understand their impact on biodiversity and mitigate, and try to get to a level that their impact is neutral, or they even have positive impact, and with that comes a lot of other groups and that are watching this, especially financial sector, insurance companies, and all of these enterprises are now focused on biodiversity as a metric, within the global sort of enterprises. And these task forces that have been formed to help organizations really develop plans. And I think from the scientific community, there is obviously the demand in terms of tools and technologies that will help this whole enterprise. Now, why is that so much of a concern? Because really it's so difficult to measure biodiversity. And the methods and approaches that we have been using have been proxy, and proxies are proxies. Let's say you look at chemical composition in order to say, hey, look, this habitat is now changed, so it cannot harbor particular type of a species, whereas what you need is a direct measure of that species in that habitat, and being able to say if it's healthy or not. And and obviously we cannot manage things that we can't measure. So this is the whole dilemma that we are facing with biodiversity analysis. And why is it so difficult? Because there's so many different methods, and most of these are labor intensive, and they're based on investigators or ecologists and field workers going in the field and trying to capture these organisms, sort them, identify them, and basically put them in databases. So there is not much automation incorporated or any type of sort of scaling at the industrial scale to be able to do these tasks. Now, what we and others have looked at, and basically as a common element of all the organisms, that's basically known in the forensic community for a long time, but now in the sort of ecological and environmental side of things, what we know is that all organisms shed some of their DNA in the environment. We call that eDNA. So if we can take advantage of this and go and find genomic regions that can determine the species diversity, then we can have a system to basically, rather than sampling organisms, we can sample environmental samples. So that's the paradigm with environmental DNA, which is based on non-invasive sampling of water, soil, sediment, or air that can reveal total biodiversity. So obviously we do need to have reference libraries of genes from known species And then we basically get these samples from water, soil, and sediment, and then we use next-gen sequencing technologies to sequence the content of these for the genes of interest, the taxonomic genes or marker genes or DNA barcodes. And then we use various bioinformatics tools to basically compare them against the libraries, and then you have identification. I don't go into the detail of the technology, but I'm happy to address those questions, later on. But this system has basically gone from the smallest scale and smallest studies to a global movement, especially when it comes to academic work and government and regulatory. So this is a map of all the studies. You see that there are lots of different data sets from different groups of organisms, and different sectors are getting engaged. As you can see, mostly the work has been done within academia and some of the government, but we are starting to see industrial applications, but various industries, environmental consulting companies, or as we'll talk about the, commercial sector are starting to utilize these tools in their workflows. Now, there are a number of things that, like eDNA, like is the pros and cons of the methodologies. But by and large, I think that the scalability and the standardization that you can have with eDNA, and the fact that one sample that can provide you information from bacteria and viruses all the way to mammals, vertebrates, that makes this system something that can become, like a universally applicable approach for a species identification. Samples are easy to collect. You don't need to get organisms and like damage the actual, like biodiversity by trapping them or sampling them. You can use various types of automation from field work all the way to robotics in the lab and sequencing and bioinformatics. Because of all of this, you have lower risk of having large number of people in the field, especially in remote locations. And then you achieve better sensitivity through the analysis, and I'll show you. And it with all that is also cost effective and time effective. Some of the examples, and sorry, some of these are got more marine angles, but like you will see that we have been using eDNA in a number of studies that are very relevant also to the mining activities, from biodiversity baseline to restoration and remediation to monitoring the impact to understanding the health of ecosystem. We can engage communities, and I'll give you an example that how you can actually use these tools for like citizen science programs and so on and so forth. In Canada, we have a unique challenge or maybe opportunity, which is basically a lot of our activities around the mining and energy sector are done in remote sites. And because of that, the challenge there is that how can we ensure that these sites, are like basically not damaging the environment, are done in a sustainable manner. And obviously it's important economically to have these activities. On the other hand, I mean, like all the stakeholders, including indigenous communities, that have these lands and waters, in their territories, they are concerned about the impact of these activities. This is a map of all the sites a few years ago with my colleagues in Environment and Climate Change Canada. We developed this sort of map for one of our bigger projects in terms of the active mine sites, and then some of the ones that have been licensed, in red, and then the hydropower dams. So this is energy and mining development, and you can see a lot of these sites that have been licensed are in remote locations. So to make the point I showed in previous slide, and they require methodologies and approaches that can be deployed quickly and at a scale in order to understand the environmental impact of them. We have been using funding, especially from Genome Canada, sort of, and various projects that we have had in systems that basically allow us to understand some of these impacts. Namely, we worked with agencies like Environment Canada and Parks Canada in Wood Buffalo National Park, which is sort of adjacent to activities of the Alberta oil sands and then some of the hydroelectric activities in British Columbia may impact as well. What we found in 12 years of this type of work is that the approach of metabarcoding or eDNA give us more species data compared to the traditional approaches that have been used by agencies and so on. This is going to give us also better power to resolve changes in the communities and understand ecosystem change. And there have been a number of publications, like the latest one came in PNAS to demonstrate the validity of this approach in a system, larger scale system of remote wetlands in the Peace Athabasca Delta in Wood Buffalo. Based on this framework, we have also as part of another Genome Canada project that just ended but continues with basically other funders and stakeholders, which is the community engagement, stream sequencing the rivers and for environmental assessment monitoring. These are local communities, many of them indigenous communities, and many of these have linkage to mining activities in these sites that we have been monitoring. You can look at the website, and if you're interested in getting engaged, definitely happy to talk about this. But as part of this, we have enforced a number of standardizations from outreach and recruitment to training and certification of local communities to sampling all the way to bioinformatics and generating reports. These are the reports that are generated rapidly after the data is generated, and we have developed a number of automated systems to help us visualize the data, analyze data. The project has a data portal that basically allows us to communicate and disseminate the information to all the partners and the stakeholders. I'm going to just spend another 5 minutes to talk about one of the projects that we actually launched with. Yep.
Sorry, Mehrdad. Sorry to interrupt. We are at 15 minutes.
Can
so
I take
if
2
you wouldn't
minutes to
mind,
show
take
this
it.
one?
Yeah,
Just 2
you can
minutes?
take another
Okay.
2 minutes, maybe, if that's okay. Just to thank you.
Okay. So this is a project that we have done with Taseko Mines in BC, and this might be interested for this audience. And basically, the idea is to use eDNA to detect mammals and their Yellowhead mine site, which is a large-scale copper mining in British Columbia. And just by sampling water, from the catchment, we have been able to determine the diversity of all the mammals in the region. Obviously, the discharge goes to the watershed, and then the DNA is in there, and we use that eDNA. And this has had a very positive impact on the activity of the mine, especially on the environmental, social, and governance. This work is done within a commercialization plan that we've had with eDNA Tech and Sika. And again, you can reach out to the folks there in the company to talk about various applications. And basically, I also want to shout out about some of the international standardization activities that we have with the eDNA, ISTF, the task force, and also the ISO working group that's been formed for this. So this is not done in an artisanal way, and it's all becoming a standard and can be adopted internationally. And thank you so much. Sorry if I went over time.
Not at all. Thank you so much for your presentation. It's, I think, very clear what a transformative technology using eDNA for monitoring biodiversity and also looking at the impact, remediation after mine closure. So I look forward to some of the questions that we're going to get after the talks. Thank you, Mehrdad. Next up on our list, we actually have Ross Orr from BACTEC with us today. Thank you, Ross, for joining us. Excuse me. Ross is the president and CEO of BACTEC Environmental, and Ross initially brought BACTEC Mining Corporation to Canada from Australia in 1997, so the company's been around for quite a few years, and was very instrumental in the success of their IPO. In 2010, the company was split into REB Gold and BACTEC Environmental, the latter delivering effective and eco-friendly bioleaching and remediation solutions to commercial operations to process and recover preferred metals and safely remove and transform harmful contaminants like arsenic into benign EPA-approved products for landfill. So today, Ross is going to be talking about bioleaching 101, and I will share my screen here 1 second. And Ross, just let me know when you are ready for me to advance the slides here. Is that visible? Oh, you're muted, Ross. Sorry.
Hate when that happens. So thank you very much. Good morning or afternoon, depending on where you are. I'm just going to run through a quick presentation on what we are doing to employ naturally occurring bacteria to replace smelting and roasting in the mining industry. We are a true ESG story. There's no greenwashing here because we can basically identify and point out all of the attributes that we bring to the table when employing bacteria. Next slide, and then the next slide. So sustainability at our core, it sounds corny, I know, but it's the facts. We employ a host of different bacteria that we use almost in a Darwinian fashion. We introduce three different strains at the beginning of the process, and 30 days in, probably 90% of the work is done by a dominant strain. We do chase arsenic because nobody else does. And the best place for commercial success is to operate in an environment where you have no competition. So we produce a ferric arsenate, which is a US EPA landfillable material. And on the back of that, we will, because we're dealing with a lot of small mines in South America, especially in the project in Ecuador, we will not buy any material that's been pre-treated with mercury or cyanide for that fact. We will also not be buying any material from mines that are not compliant with local environmental standards as applied by the government of the country. So it's going to force a lot of the miners, and there's over 100 mines in this area of Ecuador, to go ahead and become compliant environmentally, which is a win for everybody. We recycle our water. There are no emissions to speak of, and of course, we like to say that bioleaching is a permanent solution. It's not a Band-Aid, like capping a tailings deposit. We actually go in and take the sulfides out and put it to bed. Next slide. That's probably not of interest to this group, so you keep going. So an interesting team led by Dr. Paul Miller, who actually resides in Perth, Australia. Paul's been in the bioleaching business for well over 30 years. David Tingy is our COO, who is a Brit, who's now a Canadian, who has worked extensively in Bolivia and other areas and Guatemala, etc. Interestingly, a note, the bottom right-hand corner is Jay Naster, who used to be the head prosecutors for the Ontario Securities Commission. And if any of you remember the Bre-X trial, that's what he did for the first year of that disaster. Next slide. So everything is milestones in mining. And of course, everything is heavily slanted towards the environmental side, i.e. permitting. So what we did was we actually went to the area in Ecuador called Ponce Enriquez, where all these mines are located, and we collected 5 samples from 5 of the bigger mines, all of which had relatively high levels of arsenic in a concentrate. And we did a bioleach test for about 6 months, and we got 96% gold recoveries and effectively 100% arsenic stabilization. Well, that's all fine and dandy, but can you make money? So we engaged an independent third party to do a feasibility study, a bankable feasibility study, and the numbers were astounding, almost to the point where people say that they can't be true. We then went after our environmental impact study, which we got done in 7 months. Obviously, the government sees a great benefit to processing this material in country as opposed to shipping 50% of the value overseas to Asia. Along the way, we picked up a nice, testimonial from Moody's, who reviewed our project and our technology and gave us an 80% actually social green bond. I mean, it's because of the fact of what we're doing in the local community. And then finally, with respect to the local community, we got a 100% support from the local inhabitants of the area where we're building our plant. So we're ready to go, except for the money. Next slide. Next slide. So effectively, when we're dealing with concentrates and tailings, these are all concentrates, even the ones that are made from the tailings. And by concentrates, if you're unaware, it's the 20% of the value in rock, whereas the 80% is the waste rock is eliminated. So you end up with effectively a concentrate. And that's what's fed into the tanks. And over 6 days, the bacteria work away on the sulfide bonds and eventually break them down. And effectively, then you just use conventional technology to to finish the process. You've liberated gold and silver for that can be recovered into a doré bar. Anything that dissolves in acid, because the pH in the tanks is about 1.5, it's quite acidic, which is why we use stainless tanks. We start adding limestone, calcium carbonate, and as the pH rises, 3 iron and 1 arsenic band together as a ferric arsenate and precipitate out. If you have other elements in there that are of value and of commercial value, then you can continue to raise the pH and knock out. Maybe the next thing would be a copper sulfate if that was what was in the solution. And then, of course, the liquid is then recirculated through the process. Next slide. So our bugs eat rocks. I mean, when you're talking to investors, you try to keep it as simple as possible, and that's effectively our tagline. And they are literally, snapping these bonds. I like to say it's like a brick wall where the bacteria are attacking the mortar, and once the mortar is gone, everything comes crashing down into solution. Next slide. So this is more specific on our Ecuador. We might want to fly through this relatively quickly. It's a terrific project. That's all I can tell you. We are committed to a phase 2, which will cost about $60 million and effectively produce about 125,000 ounces of gold a year. We have a 12-year tax holiday, which is very attractive. And it's because we brought biotechnology to mining in Ecuador. Next slide, please. So we're out looking right now for $20 million in financing, that's US dollars, 17 for the capex for the plant, and then 3 million to pay for the upfront purchases that we'll have to do with the miners, who will arrive every day with 50 tons of material, grading roughly 2 ounces of gold. And we are in various various areas of discussion with other groups. There's probably about 10 groups looking at it right now, and hopefully we can close this sooner than later so that we can get going. Next slide. So why Ecuador? Horrific arsenic. I mean, these people are going to mine no matter what, because it's how they make a living. What we try to do is come in and get them to do it as cleanly as possible and try to stop them from dumping everything else into the rivers that ends up in Peru, which is not going to, it's going to lead to a longer term problem between the two countries. There's no question about that. Access to hydropower. It's just sort of the perfect storm for us in this particular area. The arsenic is really what attracted us there. The willingness of the people the miners to sell to us, because the Chinese recently put an import tax on high arsenic gold concentrates going into the country, which literally knocked the purchase price down to 45 to 50 cents on the dollar for the gold values that the miners are getting. And of course, they're not happy. Next slide. This is the investment protection agreement that we signed recently. Apparently, I'm doing another signing at PDAC next week with the Minister of Production. Effectively, what they've said is you get a 12-year tax holiday as long as you build the second phase, which we fully intend to do. And there's enough material to supply a plant of up to 250 tons a day. It also gives us an independent arbitration site so that if there are issues with future governments, we will not be subject to a court like they were in Panama recently, where 7,000 people were kicked out of a job for. Anyway, I digress. Next slide, please. So that's phase 2. Keep going. This shows the sensitivities to the project. And as you can see, at $2,000 gold, our after-tax profit is literally almost our capex for year 1. Next slide. So this is an interesting, I'll just quickly skim over this. Pyrrhotite tailings in the Sudbury Basin. Next slide. Pyrrhotite is a low-grade sulfide mineral that's very, very difficult to deal with. It tries to oxidize all the time. So if you leave it out in the open, it'll combust. So the pentlandite was the sulfide of choice. The pyrrhotite, they either put it into lakes to stop it from oxidizing, or they buried it. The estimates are anywhere between 80 and 100 million tons of this material. Quickly, what our goal is the bioleaching is not the tough part, because as I said, it's going to try to oxidize on its own. what we're trying to figure out is what form of iron can we generate? Because it's about 60% iron pyrrhotite. Can we put that somehow through a reduction process and pelletize it and sell it to Algoma, who are only, 100 miles west in Sault Ste. Marie? Obviously, nickel cobalt as a home, because everybody's looking for that for battery metals. And then the elemental sulfur that we'll produce, we're looking at different things, whether it be selling it to the sulfuric acid industry, or maybe even looking at the fertilizers stuff. Next slide, please. And so we are working very closely with Dr. Nadia Mikachik from Merarco in Sudbury. Nadia arguably is the best known, I should say, hopefully I'm not stepping on somebody's toes here, best known bioacademic in the mining space. How's that? And so she's running the pilot plant that's running right now, and hopefully by June we'll have some results as to what we can do with the parts. And obviously the sum of the parts, we hope, is worth more than the whole. Next slide. Is that it?
Yep, that should be the next slide. Do you want me to go to the next one?
Yes, please.
Okay, sorry.
So it just been, okay, you don't need an investment summary. This is not about investment. So that's my story. The appendix just shows a schematic of the various, what a bioleach plant looks like.
I'll put that up for a moment just in case anyone's interested. Thanks, Ross. Appreciate it. That's such an interesting story. I mean, I hear a lot about bioleaching, but it's really cool too to hear about sort of the economic and the environmental impact you're having, as well as sort of the valorization of other products from the ores that you're bioleaching. So that's really fantastic. Okay, one second here. I'm going to stop sharing. And up next, we will have our last presenter. And Cassie, once again, thank you so much for coming in last minute. We appreciate having you today. So Cassie is a science communicator with Kunki, a bioinformatics company that's based in BC. She's also a PhD student at USask. And today she's going to be talking about leveraging the power of microbial technologies for the benefit of the mining industry. Over to you.
Thank you so much. So I'm going to try and share my screen. We'll see if Zoom wants to let me do that. Oh, I do have to open system preferences, apparently. I sent you my presentation right before we started here. Is there a chance that you have that handy, or do you mind if I just hop off for a quick second?
I can get it right now. Okay, just one second here.
Sorry about that.
No, that's okay.
This computer.
No, it's all right. This is always when the technical issues happen, right? Is when you're about to present. It's law. One second here.
The delay.
It's all right. It's okay. And again, thank you. I mean, I didn't ask you for your slides in advance because, again, you came in very last minute and we appreciate that. So thank you, Cassie. One second here, folks. Sorry for the delay. Okay.
Thanks for going through the links to pull that up for me. I
No
think it's
problem.
in a Google link, so I'll make sure that I'm
Of course, now my computer's freezing. Why don't you tell the folks what mmap is while I'm pulling up your presentation?
Not a problem. So I'll do my little introduction here. So yeah, thank you so much for having me here today. This is kind of a last minute request. So I've brought with you a presentation that I've taken to a couple of mining conferences. It's intended to be just a little bit more narrative and story forward to talk about mmap and what we feel is the future of microbes in mining. So my name is Cassie Appelt. I am a science communicator at Kunki. And at Kunki, we're bioinformatic consulting leaders. We've worked in broadly diverse sectors of industry, from healthcare to mining to wine and agriculture. And we've worked with high-profile clientele like NASA, Nestle, and major universities like Stanford, Georgetown, UBC, U of A, on work that ranges from anything like trying to understand the potential for life on other planets, to how the best wine is made, or even how microbes in the ocean are affected by climate change. So we kind of do a little bit of everything, and we really love what we do. Recently, we've been working on a little something that's partnered us with mining giants like Teck Resources and Rio Tinto. And this project constitutes the largest investment in natural resource genomic bioinformatics in the history of the sector. And it's intended to support an industry in current need with immense desire to utilize genomics in the future of their operations. So I'll be talking to you today about the mining microbiome analytics platform and the future that we see for microbes in mining and offering mining. But before I launch too far into that, I have a little story that I want to take you through. So I will need the presentation now. Oh, perfect.
Okay, so forgive me
Very
because
well
I've
timed.
never used Google Slides. Is this
Oh
working?
goodness, we will see how this goes. I haven't
we're
used Google Slides for this either, so it might be interesting. That's all right,
Just-
we'll roll with it.
You just tell me when you want me to switch. Over to
Please
you.
advance the slide. Thank you so much. So before I talk to you about the mining microbiome analytics platform, I actually want to talk to you about trains. So in the 90s, the Toykato Shinkansen in Japan had a problem. It was a lucrative project that hoped to connect urban centers via high-speed rail, but it was plagued with noisy high-speed trains. And not just a little noisy, the overhead wiring that kept the trains connected to its power source could be heard rattling in the cabins. And the air resistance that the locomotive had, traveling like 300 kilometers an hour through tunnels, made like a deafening commute, but it also caused a sonic boom when it ran through tunnels. And this could be heard for like more than 4 football fields away from the tunnels that they were traveling through. So ultimately, these high-speed trains were reaching limitations in engineering to design something that connected very large centers a very large distance apart with effective and comfortable communication or transportation. Can you please advance the slide? There we go. Oh, it's not bad. So an engineer working for the railway was an avid bird watcher, and he recalled a little bird called the kingfisher that was quite adept at diving at high speeds, hurtling through the water to catch its dinner. And with the shape and the utility of that little bird's beak in mind, the engineer redesigned the front of the bullet trains. Can you advance? Thanks. So now they had this tapered design that mimicked the geometry of the kingfisher's beak. So this solved the air pressure problems that the trains were having through tunnels. Advance, please. But this wasn't the only bird used to design the 1997 500 series bullet train. The pantograph vibration problems, which is that piece that keeps the train connected to its power source, was solved by shaping it to look like an owl's face, which is concave and absorb sound. Body of the train was also redesigned. Please advance. And it was designed to look like the body of an Adelie penguin so that it could stream through the air more effectively. Next slide, please. Modeling fixes to human problems after naturally occurring solutions isn't a new concept. Biomimicry is a really normal source of engineering inspiration, and further examples in our everyday lives include anything from Velcro technology that we've designed after burr seeds, urban planning software rooted in slime mold growth patterns, green building material made from fungi. We make less painful needles when we model them after the tips of a mosquito proboscis. We make more effective wind turbines based on whale fins, and we make better water filter designs like the microscopic structure of a diatom. So we make better versions of essential tech when we observe natural examples where it's done better, from human health to transportation. And sometimes the absolute best solution is just let nature do the job for us. And we let microscopic cellular organisms do anything from creating insulin for diabetic treatment to biological fuels. Next slide, please. Speaking of uniquely human problems, we're living at a time where the demand for rare earth metals has never been higher. Our need for mineral extraction is outpacing what physical and chemical mining processes can deliver. Large ore deposits are harder to find, and declining resource quality creates higher costs and emissions during refinement. So we're reaching limitations in our ability to extract what we as a society need to diversify our energy sector. Next slide, please. The work of microscopic organisms, or microbes, in mining has promise. Can you please click next? Thanks. Microbes are already perfectly designed for mining environments. They live in acid mine drainage. They can remine mining waste. They eat methane, create copper deposits, help control dust, and they're early and reliable environmental indicators. And I think we've seen a lot of that already today with the people that presented before me. So they're already being used as greener and more effective alternatives where other industries have reached their limits. Next slide, please. We'll see if this video works. Microbes are everywhere. They're on your computer screen. They're in your digestive system. They're across the planet. Your body is made up of more microbial cells than it is human cells. And microorganisms are inexplicably connected globally. So this is their planet, and microbes of all varieties are essential to life living on it. Next slide, please. These tiniest miners have found billions of years worth of success adapting to environments that other organisms just can't, and they really haven't let mining stop them. They're the most plentiful beings on the planet, eight orders of magnitude more plentiful on Earth. Next slide, please. Than the number of stars in the known universe. That's a lot of microbes. So how do we harness their capabilities then and put them to work on mine sites? How do we use them? First, we need to know them, and the way that we get to know them is through their genes, their DNA. Next slide, please. So the good news is that the cost needed to perform DNA sequencing to identify microbes has gotten considerably cheaper, faster, and more sophisticated in the past 15 years. It took us 13 years and billions of dollars to write the first draft of the human genome. In 2007, it still cost about $13 million to sequence a genome, and now it's about $600. So companies that sequence your DNA and interpret your DNA are worth millions and hundreds of millions and sometimes billions of dollars. It's really big business. But getting to know a microbe's DNA is just the beginning to understanding microbes. Can you go to the next slide, please? Thanks. Did you know that you're more genetically similar to a banana than some microbes are to one another? That's crazy. At least I can see here, and I can't because I actually can't see anybody. But if you had your cameras on, I could tell that we're not just talking a tropical fruit here. We can't do that with microbes. We can't just look at them with our eyes or even under a microscope and really know who they are, what they're doing, and how they're working together in communities and how they live and thrive on a mine site. Next slide, please. The reality is that developing and implementing microbial solutions in a mining environment, it takes time, it takes a lot of money, and it requires a considerable amount of expertise. It takes a huge amount of work and a lot of data to understand how microbial communities change with time, how they respond to mining processes, and how they're different in different environments. It takes layers of that data to understand how those factors interact, and it takes close collaborations between scientists, mining companies, bioengineers, and more to really complete that process end to end. Next slide. Thank you. So the mining community saw so much potential in the idea of solving these challenges that we came together as a consortium to create the Mining Microbiome Analytics Platform and turn it into its own company. We've partnered with Teck and Rio Tinto, some of the biggest names in Canadian and global mining, in the development of MMAP, and we're working with 3 additional mining companies for alpha testing this year. We have fantastic collaborators in the mining space like Alonia and BGC Engineering, Genome BC, and the Center for Excellence in Mining, just to name a few. Next slide, please. So the Mining Microbiome Analytics Platform is the tool needed to solve mining problems using microbes. We don't want microbial neighbors to ever just be strangers, and we want to make that introduction. Microbes are already at mine sites. They're already perfectly suited to do work there. Next slide, please. For example, and we've heard a bit about this today, 20% of the world's supply of copper is already being extracted using bioleaching. And that's a process that speeds up normal microbial work. Like we've just learned, microbes can extract copper as a liquid from rock, and then we can use electrochemistry to revert it back into a solid to collect it. Biomining like this is cheaper, it's greener, and it has higher efficiency than traditional extraction methods, meaning that we get more from lower grade deposits. This has been done with copper for decades, but it's also being applied to biomine things like uranium and gold. At Kunki and MMAP, we don't want to stop there. Next slide, please. From a prospecting standpoint, bioprospecting for mineral deposits is cost-effective, it's time-saving, and it's minimally invasive alternative for pinpointing things like kimberlite deposits, for example, under tens of meters of earth. Notably, this work here with kimberlites was done in northern Canada, where there's a great mineral potential for other metallic deposits existing in terrain that has thick overburden. So microbes can overcome access barriers in these and other geographical locations, making for fast, affordable, and accurate prospecting possible in remote or hard to reach areas. Next slide. Microbes can improve mining processes during all aspects of the mining life cycle, from on-site chemical synthesis, next please, to dust control, next please, contaminant removal and more, next please. Thank you. With MMAP, you really don't have to stop there. So we're currently working with clients on projects related to tailings stabilization, sustainability and environmental impact, and so much more. Can you go to the next slide? Thanks. So don't wait to get involved with MMAP. We have a soft launch date in April, and we're already onboarding customers. And if you're in the mining sector, if you're interested in genomics as it relates to mining, or you're just genuinely really excited about microbial genomics like we are at Kunki, we're always happy to have a chat. Thanks so much for having us here to talk a little bit about MMAP and some of the work that we're doing, trying to get microbes working at mine sites.
Thank you so much. That was so interesting, Cassie.
Thanks.
You pulled that together last night. Oh my goodness. That was fantastic. And it's so interesting to me. I mean, you think about sort of the role of the microbiome in so many nature-based processes, but to see all of the work you're doing, it's kind of amazing. I like wrote down one-stop shop for microbial processes. It's pretty fantastic. Thank you for that. So now I'm going to hand it over to Nell. We're going to have an open panel discussion, and so we'd be happy to take any of the questions that you have. Of course, we've got a couple that we can kick off with, but if we've got any in the chat, I don't know if we have any at the moment.
Yeah, so I've been keeping an eye out. I don't see any there currently. So I guess we'll give folks a couple seconds there to see if there's anything that they want to ask. I mean, that was a lot of information to take in the last 40 minutes or so. So thank you so much to all the presenters. I guess maybe oh, there we go. All right, surface prospecting for kimberlite is interesting. How deep and rich would the deposit have to be?
I think that's probably for me.
Yeah,
So
sorry.
this was a really recent Nature publication, and it doesn't come from our group. They were able to pretty reliably predict where kimberlite deposits were based on they had some data. They were then using that kind of blindly in order to then try and see if they could test their models. And they were able to find it under tens of meters. And I don't quite recall specifically because it's been a little bit since I've read this paper now. But they were able to predict it somewhat close to the deposit as well. So it wasn't as easy as taking a ground sample and being like, yes, somewhere in 100 kilometers, we've got kimberlite. But as you get closer, there were more reliable evidence based on microbiome changes that perhaps there was kimberlite there. So it's a model that's still being developed. And with NMAP, it makes it a bit easier to try and develop those things.
Awesome. Thank you so much, Cassie. That was a good question. All right, microbes in mining. How clean, accessible, and affordable is this method? And I assume that one's for Cassie as well.
Now, I'm not going to pretend to be an expert in all things microbe. Kunki does a lot of work with microbes, and I am the science communicator. And so if there are other panelists here that want to speak more to the efficiency of using microbes in mining with very specific applications, because we've got some people here doing some really incredible stuff with eDNA and also with microbes, please feel free to jump in. But I do know that working with microbes, it can really range depending on what you're wanting to do and what type of process that you're trying to improve on. Creating something in order to work with bioleaching, if you want to be bioleaching copper, there's a lot of literature that talks about how incredibly much you can reduce the copper, or sorry, not the copper, the carbon imprint and the output and the emissions rates associated with mining just by switching over to doing something like bioleaching instead. Also, some of the work that we do right now with our consortium members and our partners during alpha testing is looking at the ability to look at microbes as early environmental indicators. So the work is super clean and it's super accessible and it's incredibly affordable. And it's a really great way to try and ask questions about what your process is doing in a way that could lead you off very, very, very early to things that are problematic, or confirm for you early on that things are not problematic. So there's a big range.
Ross, do you want to weigh in on that from the bioleaching perspective?
Yeah, part of our feasibility study, that's exactly what we're trying to do is, determine the actual cost per ton using bioleaching to process the material that we're looking at. In the case of Ecuador, we have it down at $212 a ton. So our margins are very easy to determine because we know we're going to pay somewhere between 65 and 70 cents on the dollar, depending on the arsenic level in the concentrate. You slap onto that $212 a ton, and that's your margin. So our margins, and again, we're not paying tax, so our margins are in the sort of 22 to 25% range. And that $212, of course, takes up everything except for depreciation and that's non-cash expenses, but power costs, grinding costs. Power is probably our biggest component. It's probably 30% of that cost is the actual power to drive the impellers to keep the material suspended in solution. So and we'll stick to that number. I mean, inflation will hit it, I guess, a bit over time, but we're very comfortable using that number when we talk to investors.
Great. And Murda, it looks like you have something that you want to add in there.
Yeah, I wanted to add, I think that that's a really good question. And I would say the part of the answer is basically to use genomics tools to basically measure the impact, of any of these biotechnologies or any of the approaches that we use for production or enhancement or. So I would say, let's say, like we can deploy an approach that we basically sample the environment downstream of these interventions, either chemical or biological or physical, and then we can measure how, for example, these microbes that we incorporated to help us, in the process of the mining and refining, are maybe getting in the environment and how they are basically impacting or not impacting, other organisms. Because obviously, what we want to do is sustainable activities, which has no like less impact on the local biodiversity and local environment. And I would say that we can use genomics tools for the whole process, for engineering methodologies that can help us use this bacteria or microbial processes. And then down the road, in order to measure their impact, we can also use other solutions from genomics like eDNA approaches and so on. So it's a whole circle, like with the tools that we have at our disposal.
Fantastic. We have a question from Heis Van Rooyen. So if anybody here is here from Alberta, Heis is actually a rep from Genome Alberta. So he's a good one to connect with. But his question is, what are the regulatory regulations around introducing natural microbial communities to facilitate bioleaching in places where they may not have been found naturally? So I think whoever wants to take a stab at that, Cassie, Ross, Mehrdad, please do.
Well, I can just speak from what we do, and that's, and it did come up with the Ecuadorian government. We have a suite of about 60 bacteria. And again, we tend to, look at the actual mineralogy that we're working on, and we can quickly identify which bacteria would be better suited for that. They tend to be in their own, I'm not going to get too scientific here because I'm just the president, right? So I'm surrounded by people a lot smarter than me. But as I mentioned earlier, it's a bit Darwinian in the process. We were able to convince the Ecuadorian government that there were no issues really of importing bacteria or other material from other countries for that matter, because bioleaching tends to operate in a fairly large envelope. I mean, we built a plant in China that treated material from 15 different mines from as far away as Greece. So the bacteria that we were using were adapting all the time to sort of get more accustomed to the material that they're working on.
I can have a quick also comment on this. I've been involved with some of the discussion with regulatory side. It's obviously more difficult when you engineer a microorganism to introduce in the environment. If it's a natural one, it's less complex. I would say that the regulatory regime of introducing these biotechnologies in Canada are, I would say, some of the more conservative ones, and in some other countries like US and elsewhere, they're less. However, I think the key issue is again to have a program to understand and demonstrate the impact. Like by nature, some of these microbes basically, once the substrate that they're basically metabolizing is finished, they just basically vanish and their community is sort of basically destroyed. But that needs to be demonstrated. There is also the issue of horizontal gene transfer between different strains of bacteria, that needs to be considered. So there is some regulation, and I must say that it's tougher when you have engineered something that is not naturally occurring. But I would say that like the tools that we have in order to understand those impacts, I think it's better to sort of convince the regulators in terms of these technologies. Yeah.
Mehrdad, we've got another question for you from Dennis in the chat, and it's an interesting question. Dennis, of course, about measuring baseline conditions over time as the natural ecosystem changes. So what if you did an environmental baseline study and the industry has to remediate, wouldn't those conditions be difficult after 50 years of climate change? And actually, I wanted to add a comment because I wanted to ask you about this too. And this kind of aligns, Dennis's comment really aligns with sort of a question I had about what's happening in the UK with this new net biodiversity gain, the net gain. And so if you're unfamiliar with it, any development after restoration needs to be shown to be 10% more healthy, diverse, whatever. And so that going back to Dennis's question, how do you confound those two things? Because it's, you know, things will change over time. So
Dennis always asks interesting questions, and hi Dennis, wherever you are, and thanks for. I mean, I am very familiar with the scene in Europe and UK because these regulatory sort of frameworks are basically more advanced in Europe and in UK. I was in London in a couple of workshops actually in the past 3 months discussing these issues. Now, what I think is really important is that understanding that here we are measuring change, and the notion of what is pristine or what is natural is a tricky one. But there are ways that we can basically try to make these measurements more, I would say, objective. One way that has been used mainly in various different sort of regulatory frameworks is the use of reference sites. So basically, you identify and designate a number of sites, and that's really why it's important to have all these conservation areas in every region, like every habitat. So let's say you have an active mining site, but then you have like I showed you the one that with oil sands, like parts of the Wood Buffalo National Park, like from the same type of habitat, same geography, same climate, and everything. It's just that some are impacted by that activity, and then you have some sites that are not impacted or presumably not impacted by that particular activity. There may be other things going on, naturally or other. So you basically measure delta. You measure the change between what's happening in your impacted site. And all of these regulations, by the way, that you're referring to, does make note of reference sites. So say that you need to identify reference sites. And now the good news is that we have a lot of these, sites. In some areas we have less. And then the other thing is that we have started to use bioinformatics and ecoinformatics to basically come up with like reference sites for areas that we may not have. Basically reconstruct a habitat, an ecosystem, and basically compare the change against those. There are a number of machine learning based methods that you train the system, say like this is the characteristics of a habitat, and then you can then look at the changes, across the space and time, and then continue to measure the delta and see how you're progressing or not. Now, in order to sort of demonstrate that you're gaining, obviously you need to show that you're getting closer to your reference site, and getting more, species or organisms that contribute to the ecosystem services in your habitat. So that's where it's going. But I'm telling you, there's a lot of unknowns still. There's a very nice area of active research. So if you're interested in doing research, this is going to be a very, I think, fruitful and useful place to research. Yeah.
All right, thank you for that answer. So we had another question in the chat. Is there any movement from governments and regulators to go beyond conventional biodiversity approaches towards eDNA for monitoring, permits, environmental impact assessments?
That's probably for me as well.
I would suspect, yes.
Yeah, so yes, there is, and there is a lot of that. I mean, unfortunately, I miscalculated my timing with my presentation, but at the end, I tried to introduce this standardization task force, for example, that we put together, and there is a website you can look at it, istef.global, IESTEF, and it's basically you will see that there are a number of regulatory scientists, from Canada, US, UK, Japan, other places, and they're very well aware of these new technologies, eDNA, advanced geospatial and remote sensing technologies is another trajectory that in combination with eDNA can be very powerful. But obviously getting them to the hands of users and making them routine, it's been challenging, but like there are good things happening right now, and we'll see a lot of receptivity. For example, I give you a good one. It's the use of eDNA in some of the activities with wind farms. Like there is some on the mining site as well. An area that has been a lot advocated by regulators is invasive species, and almost all of the industrial activities, they do need to demonstrate if they're introducing or changing the dynamics of invasive species in the region, and their impact also on endangered species. So there is a lot going on there.
Great, thank you. I'm going to jump around the Q&A a little bit here. So there's more of a comment. I'm a geologist. I'm interested in genomics in mining because of the negative impacts of chemicals like mercury used in mining on the environment. So I guess I'll put that one out to the panel and see what folks want to chat about on that one.
Well, I think as I stated before, I mean, our answer to that problem is just to avoid it 100%. It's a sort of carrying a big stick and a carrot. If you want to be paid more money for your product, then give us the best product you can provide. And that involves not pre-treating it and trying to steal the easy gold, but thereby contaminating the material that's going to be going into the tanks of the bacteria. So for us, it's not only an environmental question, it's an economic question. If you're going to kill your workforce, you're not going to be buying that material. So, and as I said before as well, we're not going to be dealing with people that are not, that don't have a valid environmental permit with the government.
Okay. All right, thank you for that answer, Ross. So there's a couple here from Dennis, and I assume I can combine them. So bacterial species is good, but do you look at gene sets that change? Would that give you better information? What I mean is that gene activities may be indicative of what the bacteria are doing. And presumably that one's for Merdad.
I don't know if I understood the question very well, but Dennis, can you turn on your, can they speak, or can they, or they should just only use chat?
Good question. Let me check.
I think we can unmute him.
Yeah.
Let me see. All right, allow to talk.
Right.
Oh, here we go.
Yeah, can you hear me now? Sorry, I mean, I don't mean to ask so many questions, but I spent a lot of time in mining projects, and what's interesting to me is when we look at bacterial species, that tells me sort of in an ecosystem, what's there. But when you're mining that data, especially in terms of metagenomics and shotgun, isn't it sort of good to be looking at gene sets, maybe in certain sites where there are certain minerals, because then you actually know what the bacteria are doing? And I know initially we were looking at 16S, but to me, gene sets for really bioprospect, that's sort of where I was going there. And are we keeping those databases?
Yeah, we have been doing that, and others too. So like, for example, you can develop probe sets or primer sets for, let's say, I have the experience on the hydrocarbon side, but same thing, it can be any other type of chemical that they metabolize. Basically, the biodiversity, local biodiversity, you can determine functionally rather than taxonomically by looking at the gene sets. You design primers for a handful of hydrocarbon metabolizing genes, okay? And then you use them to PCR up and do metagenomics, and you will see what's going on. Or you go full metagenomics and then harvest the data for the genes of interest for the chemicals that you're interested in. Both of these are happening already. There are a number of projects I know that basically are using that for bioprospecting, for finding microbes that then can be used for various types of chemical processes, like the one we heard about bioleaching and so on. So it can be used, from that perspective. Yeah, for sure.
Cassie, please. Yeah.
Yeah, can I just hop in here? So I just wanted to respond to the question of well, are we using gene sets? And I don't mean to, come in just personal between colleagues, but are we using gene sets? Who's using gene sets? Are we keeping that data available somewhere? I just wanted to say very quickly, at MMAP, we don't really do the bioengineered solutions portion of it, but what we are is a platform that gathers data. And so part of what we're offering is the world's first and largest data commons for mining related sample data. And part of what our company is and will be, because we'll be launching in April, is just the automated sample to sequence processing of sample data. And so the automated bioinformatic pipelines that do exactly that, give you a list of ASBs, what is in these samples, who's in these samples, but also the gene stats needed to start making, conclusions about what are they also doing. And so that's an automated process with MMAP. And I just wanted to respond and say it's something that's not necessarily widely available in mining right now. And I think there's a shift as a whole in genomics in mining related, industries to want to fill that gap as microbial solutions are starting to be implemented.
Yeah, that's fantastic. It's such a cool platform that that's being developed. We're going to do one last call for questions before we wrap it up.
Quick note though, I had someone ask in the chat if the presentation is recorded, and yes, it is. So we will try and make that available afterwards.
Great. Any final comments, closing comments from our speakers that you'd like to add?
Thank you so much, guys, for organizing it. And I think this would be good to, one of the things that I always recommend, as I said, I've been sort of in and around mining community for over a decade, and we have had a number of genomic sessions, is that the technology is evolving so quickly, new solutions, new platform, new companies bubbling up. So it's always good to renew these discussions every year or every couple of years, especially with genomics. It's so rapidly evolving. So I like to propose that this become an ongoing, discussion, and then where, we can get diverse groups coming in and talk about their innovation, but also get the other thing that would be fantastic to hear is that what are the needs in the mining sector? What is their, let's say if you're a mining company, what is the type of solution that you imagine having, and then we as the developers of the genomics community and the scientists and technical people can think about this, from perspective of commercialization and innovation. So it would be good to have, discussions and hear folks from the mining side, in terms of the challenges.
Great.
We're really hoping. Go ahead. Sorry.
You go right ahead, Kristen.
Well, all I was going to say is I think, that is the goal. I think we're really hoping that this webinar sparks the interest and that there is more ongoing conversation. So I'm hoping there's some really fantastic follow-up that comes to all of you on the panel and to us as well. So, it's a great comment. Thank you. Sorry, Janelle, go ahead.
Yep. No, all good. So, I guess that we'll wrap up now. But first of all, I just want to say how amazing that, this turnout is. I'm so glad so many people are interested in genomics and mining. Just seeing the potential of innovative solutions to industry challenges in the mining sector is always phenomenal. The session was an idea that myself and Kristen started chatting about probably a year ago. So to see it actually come to life with speakers and attendees from all across the country is just absolutely incredible. So we really do appreciate everyone taking the time to attend the session, and a special thanks to our speakers, Murdad, Ross, and Cassie. We really hope everyone found the session interesting and that you have some thoughts about how these approaches could be integrated into what you do. So I'll turn it over to Kristen.
Great. Again, just before we go, one last thing we want to mention. Genome Atlantic and CNA/MICA will be partnering on an initiative in the coming months, so please stay tuned. The goal of the partnership is really to help fund or support Atlantic Canadian companies that are interested in dipping their toe in or incorporating some of these genomics or biotech approaches into their business. So it could be anything from running a small pilot project on site to hiring a consultant to explore whether some of these options are feasible for you. So stay tuned. We're really excited. It will be launched once the agreement is signed, but we will be doing an announcement once that's funded. So thank you everybody for your time and have a wonderful day.
Thank you, everyone.
Thank you.
Bye.
Cheers.